In this lecture, we're going to take a look at our last major class of biomolecules, lipids. Now, when you think of lipids, you might think of fats, right? So fat is one of the main energy storage mechanisms in our body. We store fat in order to save that energy for long-term use as our body needs it. Or you might also think of lipids and membranes. So lipids, of course, make up all of the cell membranes or the organelle membranes within our body. Lipids have a wide range of different roles. So in addition to this fat and membrane-based role, we also have a variety of different signaling lipids, and then also many other minor roles of lipids in different places as well. We also see a similar diversity of structure, right? So in most of our other biomolecules, we had kind of a defined set of monomers that were then built up into varying polymers. With our lipids, we have much more diversity in terms of what our structures look like, right? We can have these long carbon-based structures of fatty acids. We can combine fatty acids together with other molecules to create some sort of almost polymeric structures. But then we also have very different looking structures that don't seem to share a lot of similarities with our fatty acids. So rather than having one specific set of monomers, our lipids are just organic molecules that are characterized by low solubility in water. So they're essentially just a catch-all group of biological molecules that are all nonpolar or hydrophobic. So there's not really any one defining structural feature besides that emphasis on hydrophobicity. So again, if we look at the function of our lipids, we see they are, of course, involved in storage of energy. They're involved in membrane structure, those two key roles that I mentioned before, but then they also play roles as signaling molecules and many other minor roles as well. Our diverse set of lipid molecules can be broken down into several different categories. First, we have complex lipids, which are lipids that contain those fatty acids. So these will be the most regular looking structured lipids with, we'll see these repeating fatty acids within different lipid structures. We can further break down these complex lipids into two different categories. So we have storage lipids that are all involved in, of course, energy storage and the fat deposits within our body. And we can have membrane lipids, which are involved in building up membrane structures in one form or another. And then we have a second main category, that is lipids that do not contain fatty acids. So of course, as you can tell, this is a very broad category that would include a variety of different lipids from sterols with large ring structures to various vitamins and pigments that will have longer carbon chain structures that are not our fatty acid structure. We're going to start by focusing on this first class of lipids, our complex lipids. Now, of course, in order to understand our complex lipids, it's important that we first understand our fatty acids. Fatty acids are carboxylic acids with very long hydrocarbon chains. Generally, our carbon chain length will vary between 4 to 36 carbons. We'll tend to often have even number of carbons in most of our natural fatty acids, and they'll normally be unbranched or linear chains of carbons. So here we see a couple of different examples. So in both cases, we have our carboxylic acid or carboxylate group at one end, and then we just have our long chain of carbons coming off from that carboxylic acid. So our carboxylic acid, of course, as we know, would be a polar group. It can also be negatively charged depending on the pH. But that polarity of our carboxylic acid is overwhelmed by the hydrophobic and nonpolar nature of our long hydrocarbon chain, right? So we have one carbon with that's polar, and then you'd have, depending on what fatty acid, you might have as many as 35 other carbons that are all nonpolar. Our fatty acids can be separated into saturated fatty acids and unsaturated fatty acids. So a saturated fatty acid would have the maximum number of hydrogens bonded to each carbon, or in other words, it would be an alkane hydrocarbon chain. Our unsaturated fatty acids have alkenes, which would refer to, of course, our carbon-carbon double bonds, right? So we say that it's unsaturated because we have less bonds to hydrogen than we normally would have if we didn't have this carbon-carbon bond. In this case, this would be a monounsaturated fatty acid, where we have a single double bond. If you have multiple double bonds, we would refer to that as a polyunsaturated fatty acid. Now, of course, when I say multiple double bonds here, I'm referring to multiple alkene carbon-carbon double bonds. Every single fatty acid, of course, will have that carbonyl double bond from our carboxylic acid functional group. With the relatively simple structure of our fatty acids, we can also use a systematic nomenclature to specify the exact structure of our fatty acid in our name. So, of course, we could use a standard IUPAC nomenclature to name fatty acids. So that would be based on the number of carbons as well as the positions of each of our functional groups. So this could be referred to as cis-9-octadecanoic acid. So, of course, our number of carbons are indicated here. We have numbers indicating where functional groups are, right? So that's indicating our double bond right here. And we have, of course, an ending indicating our carboxylic acid functional group. So this is our systematic IUPAC name. The systematic name is useful for specifying the structure of our fatty acid, but of course it can be a little bit long and inconvenient. So we'll often see shorter common names used. So for our fatty acid here, this would be called oleic acid. But oleic acid, of course, doesn't tell us what the structure of our fatty acid is. So we've also developed a shorthand nomenclature to indicate the key details of our structure. So we'll just see a few key numbers here together with symbols. So our first number indicates the number of carbons here. So we have an 18-carbon fatty acid. The second number here indicates the number of alkene double bonds. So this is a monounsaturated fatty acid. And this delta symbol and our 9, so this is referring to the position of our double bond here. So we call this a delta numbering. If we count from the front of our carbon chain, where we have our carboxylic acid, we have our double bond at the 9th position. So delta numbering is just numbering from the front of our carbon chain. There's also a separate omega numbering system that would number our alkenes from the back of our carbon chain. So if we're using omega numbering, we would refer to this as 18,1 omega 9. So we, of course, still have the same number of carbons and the same number of double bonds, but now we're counting from the back. And what we see here is that it actually also happens to be at the 9th carbon, whether we count from the front or the back. We can use the same nomenclature to describe more complicated fatty acids as well. So here we have a longer fatty acid chain. So notice we have 20 carbons here. So this would be a 20-carbon fatty acid. We have 5 different double bonds, again represented right here next to our colon. And then we have a number indicating the position of each of our double bonds. So it's at the 5th, 8th, 11th, 14th, and 17th carbons in our fatty acid here. So this, of course, would be polyunsaturated. We would also have a common name to go along with this. So this is eicosapentaenoic acid, or EPA. Now, the name EPA might be familiar to you because it's an important dietary fatty acid. So if you're at all familiar with nutrition, you might know that omega-3 fatty acids are essential nutrients. We actually need to have those fats within our diet. So fats like ALA, DHA, and EPA are all various omega-3 fatty acids. We can't synthesize these, so we have to include them in our diets. So what do we mean when we refer to omega-3 fatty acids? Well, that, of course, is referring to that omega numbering. So something like our oleic acid shown here is not an omega-3 fatty acid because it has a omega-9 double bond, as we saw with our earlier numbering. If we compare that to our EPA here, so again, our omega numbering is starting from the end of our carbon chain, not our carboxylic acid, and we see our first double bond is at the third carbon, so this would be an omega-3 fatty acid. So that omega-3, or you might have heard of omega-6 fatty acids, it's just about where our double bond is positioned relative to the end of our carbon chain. Using this basic nomenclature, we can describe the variety of commonly occurring natural fatty acids. So first, with this table here, we have our saturated fatty acids. So these would just be varying in our number of carbons here in our carbon chain. And of course, all of these have a 0 since they don't have any alkene double bonds. We also see our systematic names here, which aren't often used because they are somewhat long and difficult to remember. And then we have all of our common names over here. The most common and most important fatty acids here are palmitic acid and stearic acid. These can also be referred to as palmitate and stearate. And of course, that's because our carboxylic acid group can be deprotonated to the carboxylate form. Right? So any molecule that has a carboxylic acid will often have kind of two different forms of the name based on the protonation state of our carboxylic acid there. So palmitate would technically refer to our palmitic acid after it's deprotonated. Here we have our common unsaturated fatty acids. So again, of course, you can notice we have our number here indicating one or more double bonds. So we have 1, 2, 3, and 4 here for our different levels of unsaturation. And we also have our delta numbering telling us where those double bonds are. So our common unsaturated fatty acids, so oleic acid is one of the most common. This is found in olive oil, for example. Linoleic acid is a relatively common polyunsaturated fatty acid. Arachidonic acid is one of the most important polyunsaturated fatty acids. So with our delta 14, this would be a omega-6 fatty acid. So what's the difference between all these different fatty acids, right? We have a whole variety of different saturated fatty acids and different unsaturated fatty acids. How does that affect their natural behavior? The first difference that we'll see is in our solubility. So the solubility of our fatty acids will change as we vary the number of carbons, so the carbon chain length. If we increase that carbon chain length, we're making our fatty acid more hydrophobic, which will then make it less soluble in water. So something that has very short carbon chain might have some solubility in water, whereas as that chain increases in length, the solubility will get lower and lower. Our chain length will also affect our melting point. So as we decrease the length of our chain, our melting point will decrease, right? This is following the typical rules that we've talked about in organic chemistry and general chemistry, right? As your molecule gets smaller, you have less of those interactions between your molecules, and so it's easier to separate those as you move into the liquid state. Our melting point also decreases as the number of double bonds increases. Now, this trend can be a little bit harder to explain, but if we look at the structure of our fatty acids, so of course, our saturated chain will tend to adopt a relatively extended conformation in order to limit steric hindrance within our carbon chain. Now, of course, it is flexible, so it won't just stay in kind of this straight line like this. It'll be moving around, but it'll tend to stay more extended rather than balled up into a tight structure. Our unsaturated fatty acids can't form this same sort of extended conformation, right? So we can form one straight chain here, but then when we get to our double bond is in a cis configuration, right? Remember, cis refers to the two bonds on either side of our double bond going out in the same direction, whereas trans would be with them pointing in opposite directions. Most of our double bonds in fatty acids will tend to be cis double bonds, and this creates this bend in our carbon chain, right? So we can have a straight portion at each end, but we always get this bend or kink in the middle of our carbon chain. So if we stack these fatty acids together, what we see is that our saturated fatty acids can pack together very nicely in a very orderly fashion, right? They all kind of look the same. We can just stack them right next to each other, lined up together. In contrast, our unsaturated fatty acids have a much harder time packing together in an orderly fashion, right? So we have this bent structure, and it's really hard to line that bent structure up with other fatty acids, whether they are saturated fatty acids or unsaturated fatty acids. So that change in our packing will also change the interactions between our fatty acids, right? If we're very orderly and packed together very well, we get a high surface area between our molecules, which allows for more opportunities to form our London dispersion or van der Waals interactions between those molecules. If we're not packed together very well, the surface area of contact between our lipids is much smaller, and so we have less interactions between our molecules overall. So then it becomes easier to separate those molecules and pull apart those interactions as we move from our more packed solid state. So that difference in packing then will explain the change in our melting point based on the number of double bonds. So if we look at a variety of different natural fats at 25 degrees Celsius, and we look at the different fatty acid composition here, so our y-axis is showing the overall percentage of our fatty acids, and of course on the x-axis we see three different fats here. The green is marking unsaturated fats, and the gray and yellow are representing different forms of saturated fat. So our gray are longer saturated fats, and our yellow are our shorter saturated fats. So the more unsaturated fats we have, the lower our melting point will be. And what we see is that results in a liquid at room temperature. So olive oil and other oils that are liquid at room temperature will tend to have a high proportion of those unsaturated fatty acids. So with our olive oil here, so you'll note we have somewhere around 80% or so of our fat is unsaturated. As we move to butter, so you see the percentage of saturated fatty acids increases significantly, and our butter then tends to be a solid at room temperature, although it is a relatively soft solid, right? If you heat butter up just a little bit, you can melt it and turn it into a liquid state. If we increase the amount of saturated fatty acids even more, so our beef fat here has more of those long fatty acids marked in gray, and it's replacing a lot of the shorter fatty acids that we have here in yellow. So those longer fatty acids will have higher melting points, and so our beef fat tends to be a very hard solid at room temperature. And if you would have to heat it up quite a bit more compared to butter in order to melt that beef fat. So our different fats and oils, you can tell their relative fat composition just based on whether or not they're solid or liquid at room temperature. So if we compare the properties of our different naturally occurring fatty acids, we can see these trends clearly. So as we move from the top to the bottom, we're increasing the length of our carbon chain. So we see that our longest carbon chain gives us our highest melting point. So we're here at 86 degrees Celsius compared to 44 degrees Celsius for our carbon chain that's half as long. We also, of course, see a change in our solubility. So we don't have a solubility for our largest carbon chains, but if you look at our shortest, lauric acid here, it has a much higher water solubility compared to our stearic acid, 0.063 compared to 0.0034. And we also see a higher solubility in benzene as well compared to our larger stearic acid. So solubilities for arachidic acid and lignoceric acid aren't measured, but of course they would follow the same general trend with them decreasing their solubility with that larger carbon chain. If we look at our unsaturated fatty acids, we can still see those same trends, although of course here it gets a little bit more complicated because we're changing both the number of carbons and the number of double bonds. So if we compare our 18,1 oleic acid with our 18,3 alpha linolenic acid, we see that our melting point, of course, decreases as we add those extra double bonds. And if we compare our arachidonic acid, you'll see that it actually has the lowest melting point of all of our fatty acids here, even though it has the longest carbon chain. Right? So that large number of double bonds actually overrules the effect of increasing our carbon chain length to make that lower melting point overall. So our double bonds will decrease the melting points, even if we have very long fatty acid chains, to below the melting points of our saturated fatty acids, or even just less unsaturated fatty acids. Now, when we've been referring to unsaturated fatty acids so far, we've only been referring to cis fatty acids. So our double bonds are always in that cis conformation with both of our bonds pointing in the same direction. So what about trans fatty acids? Well, trans fatty acids, of course, would be fatty acids with our unsaturated double bond in the trans configuration. So notice how our two bonds are pointing off in opposite directions from our double bond. Trans fatty acids aren't naturally formed, but they can be formed through various food processing mechanisms. So partial dehydration results in trans fatty acids forming from unsaturated fatty acids. So this is done by food processing companies in order to try to increase shelf life and stability of oils. So of course, as you're changing that trans double bond, so it will change how the fat metabolizes, but it also changes the physical properties of our fat. Right? So you'll notice right away that with that trans fatty acid, we're now able to adopt a more extended conformation. We no longer have that kink or the bend that's created here by a cis double bond. So we'd expect then that trans fatty acids would have higher melting points compared to cis fatty acids. So this higher melting point can be helpful for increasing the shelf life and stability of our fatty acid, but of course, our trans fatty acids tend to be much worse for us from a nutritional standpoint. So of course, a lot of areas have banned trans fatty acids in foods, and companies have moved to try to limit the amount of trans fatty acids that they're placing in their food products. So how are these fatty acids actually stored in our bodies? So for the most part, our fatty acids won't be stored as individual fatty acids. They'll actually be linked together in the form of triacylglycerols. So glycerol is our triol shown here. So we have a 3-carbon chain with 3 alcohols, 1 on each carbon. And we can react this with our fatty acid using an esterification reaction. Right? So carboxylic acids can react with alcohols to create esters. Since we have 3 alcohols, we can link 3 fatty acids on here to create our triacylglycerol. You could also have 1 or 2 fatty acids linked on here to create a monoacylglycerol or a diacylglycerol, but our most common fat storage form is the triacylglycerol with 1 fatty acid linked to each alcohol. So you can notice our ester functional groups linking each of these fatty acids onto our glycerol molecule, which is highlighted here with our orangish color. Our triacylglycerols are defined based on the melting points. So if we have more saturated fatty acids, will tend to be more solid. So those will be characterized as fats. And if we have more unsaturated fatty acids in our triacylglycerol, we refer to those as oils, and they would be more liquid. This triacylglycerol, or also called a triacylglyceride, will then be used as our energy storage in our body. Fats are very efficient and useful for energy storage in a variety of different ways. So first of all, our fats actually carry more energy per carbon. So as we're producing energy from our carbons, we're of course oxidizing them to carbon dioxide. Now, if you're starting from a sugar, all of your carbons will have alcohol functional groups. And so you're only oxidizing from that alcohol to your carbon dioxide. In a fatty acid, most of your carbons start as alkane carbons, right? So we just have our hydrogens attached to a carbon, along with, of course, the bonds to other carbons. So this is a more reduced state for our carbon, and so we'll be able to produce more energy as we oxidize it over more steps to get our carbon dioxide. The second key advantage of our fats over polysaccharides is that our fats carry less water per gram, right? So because they're nonpolar, they'll tend to just cluster by themselves and exclude water from our fat storage. On the other hand, when we store sugars, they'll actually have water bound up with those sugars, forming hydrogen bonds to our sugar molecules. So our sugar molecules then will always be stored with water, whereas our fatty acids are stored without water, just by themselves in fat droplets. So because of this, glucose and glycogen are better for short-term energy needs, right? So since they're water soluble, they can more quickly and easily be accessed and then transported around the body, whereas fats are much better for long-term energy needs, right? So they're much more efficient storage. So you can store more energy in fat tissue, but it is a little bit harder to access that fat and get it delivered to the rest of the body. So of course, our fat cells then act as large energy stores within our body. So this is what fatty tissue would look like. You end up with these very large fatty droplets represented here with these white areas in this microscope image. So the white are large areas of just triacylglycerols stored to provide that long-term energy storage for our body. The red here notes the nucleus of the actual cells that are storing all of our fat here. So these are the adipocytes, and then our adipocytes will have these very large droplets that are actually occupying most of the space of the cell, just storing those extra triacylglycerols there for our body to use. So as we see here, our fatty acids are a very important source of energy for our body. But again, that's far from the only use of our fatty acids. Those fatty acids are also found in our other complex lipids, including our membrane lipids. So in our next lecture, we'll take a close look at how fatty acids are involved in building up membrane lipids, and then how those membrane lipids work to form the structure of our membranes.